Hello, everybody. In this next lecture, we are going to talk about prokaryotic cell walls. So let's get right into it. By the end of this module, you should be able to compare and contrast gram-positive and gram-negative cell walls and compare L forms to mycoplasma bacteria. And so when we're thinking about the structure and function of cell walls, the main thing they're going to do is they're going to provide this like rigid structure and an extra layer of protection. Now, cell walls are made out of a carbohydrate, polysaccharide. Basically, it's a mixture between carbohydrates and proteins or amino acids, and it's called peptidoglycan. And so peptidoglycan has these repeating NAG and NAM subunits. NAG is N-acetyl glucosamine, and NAM is N-acetylmuramic acid. And archaea use S layers and sometimes pseudo peptidoglycan, but we will not talk as much about that. And so when you look at the cell wall structure, like I said, we have these NAGs and NAM subunits, and you can see that they are very similar in structure to glucose. So here's our glucose molecule on the right-hand side, and then we have our NAG and then our NAM. And so these two molecules are similar as well. And so basically these are just a type of carbohydrate. Now, one of the ways that we are able to distinguish between different types of bacteria, and this is actually becomes fairly important for clinical purposes, is by classification of their gram status using the gram stain, which is based off of the microbe's cell wall. So we'll talk a little bit about that here and a little bit more in another lecture. Now, the gram-negative cell wall is fairly thin, and we can see that in this right-hand side. It's actually going to be located between a inner and outer membrane, and then within this kind of periplasmic space here. And the outer membrane, as we talked about in the previous lecture, has these LPS, this lipopolysaccharide. Now, the gram-positive cell wall is much thicker. While this is usually just maybe one or a few layers of peptidoglycan, the gram-positive cell wall is going to be several layers of peptidoglycan. So it's going to be much thicker, somewhere in the 20 to 80 nanometers. Now, they have no outer membrane. They just have this inner membrane and the cell wall. And comparison to the gram negatives, which have the inner and the outer membrane and a thin cell wall. And so when we do the gram stain, we can view the uh, bacteria under the microscope, and the purple cells, which are these dark purple cells here, those are going to be gram positive, and then these pink or reddish cells are going to be gram negatives. Now, one thing to kind of keep in mind is that pink can sometimes look kind of purpley. And so in certain places on here, at least in my not super artistic eye, some of these pink areas look kind of purpley. And the important thing is to really notice the difference between the pink, which is the lighter color, and the purple, which is the dark color. So purple is usually pretty purple, and pink is maybe all over the place, at least in my experience when we're in the lab. All right. So I don't expect you to know all these things, but this is kind of a fun little chart of medically important gram-negative bacteria. Notice that a lot of the bacteria over here are in gram negatives just appear as single bacilli. And so that's on the right-hand side of this chart. And this is where you're going to find your Enterobacteriaceae, which includes E. coli and Salmonella and Shigella, and Yersinia, which is the plague, and several other organisms as well. Then there are some others that are maybe of interest, we've got our gram-negative cocci, which include our Neisseria, which are going to be things like gonorrhea and meningitis. And then we've got our Treponema pallidum, which is what causes syphilis. And then we have Vibrio, like cholera, which causes cholera. And then Vulnificus, which is like a flesh-eating bacteria that you usually get from seawater. Here's examples of um, our different gram-positive clinically important organisms. And so these, of course, are going to include the acid-fast and non-acid-fast organisms. We'll talk more about acid-fast um, a little bit more in the lab lecture. And then you're going to have some important organisms. Many of these you'll be at least somewhat familiar with, like Staph aureus, which is what causes a staph infection. And then we have things like Streptococcus pyogenes, which is what causes your strep throat. And then we've got our Clostridium tetani, which is tetanus. And then the mycobacteria, which includes Mycobacterium tuberculosis and Leprae, which cause tuberculosis and leprosy, respectively. Now, oftentimes gram-negative species are harder to kill with different chemical agents than gram-positive species, and this has to do with that kind of with that cell envelope structure. And so, and if you look in the outer membrane of the gram-negative cartoon here, we see these porins, like this guy here. And these are non-specific channels. And we'll talk a little bit more about different kinds of transport channels in another lecture. But what they do is they allow things to non-specifically pass through that can't just diffuse through. And that includes uh, mostly like amino acids and vitamins and things that can be used for the cell. But they do exclude larger molecules. And oftentimes, um, some of the chemical agents that are used for controlling bacteria are larger molecules. They're not going to fit through that porin to get access to, say, the cell wall. So if you wanted to use a penicillin to kill an organism, it's going to have to get to the cell wall to break it down. And if it can't get in past this outer membrane, then it's not going to be very effective. It's not to say that there aren't ways for those kind of cell wall effective antibiotics to be functional. It's just that you have this extra layer of protection for these organisms, which helps them survive some of these um, antibiotics. And so when we think about gram-positive cell walls, there are some pros and cons, right? So one of the bigger cons from a environmental perspective or an ecological perspective, evolutionary perspective, is that the thick peptidoglycan is just exposed. And so these different sorts of compounds that can potentially affect the outer membrane are going to have direct access to, or sorry, that can directly affect the cell wall are going to have direct access to that cell wall. Um, but so that does have pros, right? And so one of the bigger pros is that oftentimes your gram positives are going to be better at retaining moisture. They're going to be more tolerant of salt and some other environmental stresses. They can be protected from mechanical stresses, so they're not as easily broken. They have these teichoic acids, which if we look back here, you can see these teichoic acids that are sticking out from the membrane through the cell wall. And these are going to help stabilize that cell wall. They help maintain the shape, and they're also involved in transport cations and cell division. And so, simply put, teichoic acids do a lot of things for gram-positive cells. And so here's a nice little chart comparing your gram positives and gram negatives. It's useful to look through and kind of be aware of some of these differences and similarities. That way, you can differentiate between these kinds of cells. From the standpoint of a lot of the stuff that we're doing in the class, things that I would encourage you to think about or be familiar with is the which one's pink and which one's purple, and also thinking about how the cell wall um, functions in for these cells, both from an infection standpoint. So how do antibiotics work, and from a growth in the environment standpoint, uh, how's the cell wall potentially aiding or impeding this cell's ability to survive? Now, I'll go over acid-fast staining quickly. This is for organisms that have these things called mycolic acids. And so they have this kind of waxy layer on the outside of their cell wall. So they're gram positives, but they come with this waxy layer. And so they don't stain very well because the stain itself doesn't make it through that wax layer. Some examples are your nocardia and your mycobacteria. And when you have a positive acid fast cell, they're going to stain pink. Um, and and we'll talk again about that in the lab lecture. And again, the reason this is an important thing clinically is a lot of things won't pass through that mycolic acid layer, making it difficult to identify them with normal methods that we use for other organisms. And then also has impact on its metabolism as well. So nutrients and gases also don't pass through that mycolic acid layer, that waxy layer. And furthermore, our antibiotics don't always do a very good job of passing through the mycolic acid layer. So the antibiotics can be less effective because they have this extra protective coating. Then there are the mycoplasma bacteria. These are a group of organisms that don't actually have a cell wall. So most bacteria have a cell wall, mycoplasma do not. They their their membranes are sterol enriched, and they oftentimes are going to be like either obligately living inside of other cells or spending a good portion of their life cycle in other cells. And they are pleomorphic, which in this case means that they have multiple shapes. So we're just showing it here as a nice circle, but they can be all sorts of shapes because that cell wall that these other cells have oftentimes are what drive their shape. When you don't have a cell wall, then they can become more amorphous and take different forms. Now, in contrast or in comparison, we have L forms, which are bacteria that normally have a cell wall, but then they lose it. And so this can actually contribute to persistent infection and make them resistant to some environmental stressors um, and other antibiotics and that sort of thing. And so, lastly, we do have antibiotics that directly affect the cell wall. We've got our beta lactams, which include penicillin, amoxicillin, stuff like that. Our glycopeptides like vancomycin and bacitracin. And so this is just a schematic kind of showing how these things work. And both of them interrupt cell wall synthesis, right? And the beta lactams are going to actually bind to the transpeptidase active site and prevent it from being able to um, make these crosslinks, while your glycopeptides like vancomycin are going to bind to the D-alanines and then they block the transpeptidase, right? So it's six in one, half a dozen in the other. Essentially, what they're doing is they're inhibiting cell wall synthesis. They have different mechanisms of action, and I'm not super worried about you memorizing the types of mechanisms of action, although it might be useful to think about what's going on, because sometimes that can come up in like the pharmacology class that the nursing students take. And with that, you should now be able to compare and contrast gram-positive and gram-negative cell walls and compare L forms to mycoplasma. I hope you enjoyed the lecture, and I will talk to you soon.